thank you. It was quicker than I expected, but anyway, so I'm going to present on behalf of Panos Alexiou, who is currently at the conference, about our core facility bioinformatics, our newest core facility. And first I'll start with this picture of what is the mission, what is our goal in the core facility, because since we don't have any actual instrumentation, if you don't count the servers and computers, we actually have just our know-how and our algorithms. And so there's quite a few things that we do, but the main thing is, of course, our collaboration with the core facility genomics and analysis. All these things are built around analyzing the next generation sequencing data. So this is what we do. And here I just wanted to show what is our goal, because we don't want to do the primary Ngs analysis, and we don't want to, at least partially, don't do secondary standard, like standard things. What we want to do in our core facility, we want to focus more personal power on especially the custom project and some nice analysis directly for researchers, and of course to educate people about bioinformatics and NGS results. But for that we need to automate these first parts, because you can't get here without the primary. And this is where we actually invest a lot of effort, time and money last 2 years. So I start with the primary NGS analysis, which actually comprise mainly of these two parts, which is building and managing the information system for the NGS sequencing and for standard analysis. We developed 4 years ago, we developed this system on our own, but luckily we get a grant, some support to build a new system, which we are now developing with a private company, actually, with a professional IT company. And we are getting close. We call it Sequoia, and in a moment I'll show you just how we envision it to work. And then other part, also big part, which we don't want to do, but we are unfortunately need to do with collaboration with ICT. We need to somehow maintain the IT infrastructure, the computational infrastructure, to actually do all the analysis and all the computations we need. And there were also some new nice move forward since we bought together with core facility genomics a new storage for sensitive data. And also this year we're trying finally to transfer our computations from our older server to new HPC main, but I'll talk about it also a bit later. So the Sequoia is actually abbreviation for SecUI, and the idea there is that you can create different types of analysis. You can register your samples as you are preparing them, as you're preparing your library. You can directly register the samples through the web-based graphical user interface. And then the manager or the laboratory responsible person in the core facility genomics can add these to specific runs. And in the next step, all this could be directly analyzed, and the results should be visualized, since you're at what type of analysis you want to actually perform or your data. So if this is successful, which I hope it will be soon next year, we can get a nice streamlined analysis, at least the standard analysis that we provide, with the end result as some nice, at least QC, but I hope that also would be included basic results of our analysis. So we can spend much less time on these standardized steps and focus more on the development and custom things. The goal here, I just mentioned it, that the goal here for this whole project, which actually we promised in the grant, is that there is going to be also the clinical specific results. That hope is that this could be used directly in the clinic to visualize relevant molecular information about the patients. So that's the part of information system for the sequencing. This is a slide about our hope or our effort to transfer to the more powerful computational cluster than the single server that we currently have. Luckily, we were able to discuss with ICT and somehow come up with the solution. They provide us right now this HDHU cluster, which is a Kubernetes cluster. There's just the illustration picture of how the dashboard of the cluster look like, where you have information about the cores. Currently, of course, it's not the exact number of the cores, but we can use approximately from 100 to 200 cores, which is good enough for what we do, fast enough. The point is why we do this, that it was real necessity last year, because with the increase in sequencing, especially with the results from Novasec, which are really huge, like 2 to 5 times bigger than anything that we got from the previous machines, the necessity for higher computational power is definitely there. And since the increase in sequencing is basically following the Moore's law in our facility right now, so doubles every 2 years, so we definitely need to invest in this part. So this is the end game result, which we are not there yet, but I hope that at the beginning of 2022, we would have a system like this. So we have our web-based GUI Sequoia, which is controlling the task runner, which is hidden behind the firewall and controls the data and the actual pipelines, actual workflows that we provide or that we manage and develop, mainly primary analysis, but also secondary analysis, and that all this is computed on this HDHU cluster. We actually use this setting right now already for this ACGT project, which is a Czech project for sequencing of the, I forgot to put Czech, but for sequencing 1,000 Czech genomes. And it's working quite nicely, even with this large whole genome sequencing data that we have. So the next part, the secondary standard Ngs analysis, which is, like, this first part is probably something that US users never see, but is nevertheless very important. This second part is something that is of interest to you mainly, because that's basically the service as it is imagined as direct service that we provide. For that, we maintain, and now we somehow consolidated the list of bioinformatics pipeline that we keep as a GitLab repository. These pipelines are all created in this workflow manager SnakeMake, which is one of the most popular programs to do that right now. And we are trying to improve all the pipelines all the time, even though we have them automated already. But still, this step to have it as a Git repository or whole consistent Git lab repository should allow us to keep even the previous version to actually ensure the potential reproducibility of our analysis even a few years back, so we can run the same analysis even when the workflow was already improved. And the other main point right now that we're focusing last year a lot is this potential, this HPC deployability for these pipelines, because we developed them before just as a local running things, and now we need to include this potential to distributed system computation. This is the list. It's not completely exhaustive, but I think like most of the main pipelines, workflows that we use is there. You can see how we are trying to constantly develop and improve our pipelines. Still, the most used service that we do is RNA-seq and DNA resequencing with the follow-up of differential expression and variant calling, which all we have now very well automatized. So most of our analysis, what we do now is automatized, but still we are trying to focus also on other more exotic and more newer types of analysis, more complicated. I would just mention this single-cell RNA primary analysis, which, to be honest, is just using of software that was developed for it. There's not much of our development, but we already provide a service to get at least the table of all the gene expressions per cell. And then the next step is quite custom analysis anyway, so we can't automate it. But we have some single cell capacity now for analysis. Yeah, lately we focused on 16S metagenomics. Yeah, transcriptome assembly, that's something we worked last year on. Okay, so now to the last part of our analysis portfolio, which is the custom analysis. And it is actually something that I would say it's our main goal, or at least for me personally, is something that I would like to do more, and I think we should do as a main thing, because that's the most added value as a bioinformatics to the institute. But unfortunately, it's quite time consuming. So as I mentioned earlier, we need to automatize and spend as little time on other things as possible. And also it needs a high level of expertise. So hopefully also, since there were some personal changes from the start of the core facility, now we're fixed with some good postdocs and some good personnel that keeps this know-how for a longer time, because when people are evolving, it's hard to keep this expertise, and then you're losing the potential to actually do all this interesting stuff. Here are the examples. There are a lot of them for custom analysis that we did and what we are potentially able to do. I just put it out there to, if somebody would catch something that would be interested in, just definitely contact us. Like recently, what we did a few times, and it's quite interesting, is this combination of proteomics and transcriptomics data. So at the end, you have one table where you have a combination. You can see if your gene expression correlates with your peptide quantity in your samples. Or the transcription factor enrichment analysis to see where your transcription factor that you are interested of your interest are actually mapping. Okay. In this context, I also, as an example, want to show 3 projects that we have been working on, just to get an idea of what the custom analysis might mean and how complex projects we can handle. So here in this first one, we analyzed for ICRC data where at the end it was all revolving around this HNRNPC gene in the context of this cardiovascular disease. But the main point was that at the end, the result was necessary to combine the RNA-seq data, so expression and as specifically not only expression but alternative splicing in the RNA-seq, and then the RIB-seq, which is something like ChIP-seq, to know where the specific where the protein binds on the gene, and then also iSCRIP to get the exact position of the binding. And all these three data sources were actually combined in our core facility. This analysis were done by my colleague, and now the paper from this is actually sent. It's not reviewed yet, but it's out there. The same thing goes for this PET-CT and whole exome sequencing combination, which is a really nice project we have with Vienna Medical University and AKH Hospital, the nuclear medicine department there, where they're doing a lot of imaging, the PET-CT imaging, and then the goal was to somehow combine these genomic features on the level of mutations in DNA with the radiomic features in the sense of size of the voxels and the intensity. And to our surprise, or at least to my surprise, it actually worked quite nicely. And there were nice results that the genomic features were actually correlating with some radiomic features. And then the combined radiogenomics feature, as we call them, were really improve the prediction of clinical outcomes, the survival of individual cohorts. So there's also the manuscript now in preparation and should be out soon. So also nice type of collaboration combining completely different types of data. And then last but not least, the very nice collaboration we have here in SATEC with the Ondra Slaby Group and Children's Hospital, where we work on this with Ondra Slaby Group for 4 years or 5 years, basically from the start of our core facility. And we invest a lot of development, or it was mutual beneficial collaboration where we developed a lot of variant coding pipelines and fusion gene pipelines, and discuss a lot with directly with clinical genetics, which help us improve our analysis. And at the end, right now, there's around 240, 250 samples already analyzed for whole exome sequencing, fusions, and transcriptomics. So also quite nice project, but this is just mentioning, and I don't want to any of you users feel that I did not mention you, because we have multiple very nice, quite large projects with multiple groups, either here in SATEC or even outside. Yes, so last part is about education. So we have 4 courses available that we organize directly or participate in organization of those courses. 2 of them are more bioinformatics oriented. One is taught in fall semester, and it's Introduction to Bioinformatics, where it should be for people who are bioinformaticians or are at least interested in bioinformatics and more mathematical details of the bioinformatics, not only the results of NGS analysis. So it actually discusses the bioinformatics algorithm and the programming. And then there is the bioinformatics seminar in the spring, which also discussed more the algorithms and available tools of the bioinformatics more than the actual results. So this is more for bioinformatics, but we also provide 2 nice courses for more oriented towards biologists. Especially this one in spring semester starts from this year, 2020. Last year, 2020, but during COVID, it was actually not very successful. I never saw any of the students. But this year, we had a nice course in collaboration with Boris, with Genomics Core Facility. And basically there, the goal is to teach people how to plan the NGS experiment, how to do the lab prep, and from our side, from the bioinformatics side, how to look and interpret, and to look at the results and how to interpret the results, because the results from NGS experiment, even processed through bioinformatics means are many times very complex, and it's not very easy to actually look into them and correctly interpret. Basically the same course, but in Czech is the same, is in fall semester with some inclusions. Also, for education, we try to do workshops and direct trainings. Unfortunately, as probably for all here during pandemic, all this was limited, so we don't have actually any new workshop last year. But right now, we have some online courses in preparation for R programming language, processing of next generation sequencing in R for biologists, and for some more in-depth deep learning, so machine learning application for biological image analysis. And then what I wanted to mention probably most here is that we also provide, and it's a big part of our work, try to provide the help to the residential bioinformaticians in research groups, be it from even before the people come here, so we can help the group leaders with the hiring of bioinformaticians, because sometimes it might be hard to assess the quality, so we can consult about the hiring. And then with the bioinformatician, we can support them with the usage of our IT structure, so we can help them to provide our virtual machines, computational power that we use. Of course, we share the know-how if we have it, and of course, we also would love to collaborate on the projects or on a specific project. And I think in all these three directions, we have some nice collaborations, even to be frank, even mutually. So sometimes even we learn many things from the research groups of informaticians. So that was the description, a bit long, but hopefully nice description of what we do and what we try to do, the overview of what our core facility is doing. And now to the compulsory slides about data policy and acknowledgements. So we share the data policy, as was mentioned, with genomics, and there's one secure central storage which is accessible to all, but the data are accessible per group. But from our side, I don't even specify here how long we keep the data, because the main part is that if there are the raw sequencing data from genomics available, we can always reproduce the analysis. So basically, anytime that your data is still there, we can reproduce what we did. But still the full text of our core facility data policies out there on the pages. Results of the survey was overall positive, I would say, but I did not want to share the graphs. So I only focused also on some critiques in the text form that were provided. So one of them was that there was demand for more single cell analysis. And yes, we want to do more single cell analysis, definitely. We are actually transferring now the expertise either from PhD Petr Taužier from Šarka-Pospíšilová's group, who did all the, who's a bioinformatician in Šarka-Pospíšilová group and did all the single cell sequencing analysis so far. So now we're in close collaboration with him to actually learn from him and share our knowledge from our side. And then we have this very nice project which includes EMBL, where we hope we're going to go directly next year and spend a few weeks to learn a lot about single cell, but also about spatial transcriptomics, which is even a newer application. Then the next question was if we can provide quantitative ChIP-seq analysis, and the answer is hopefully soon, because right now it's in development. I asked Martin, and he said, I don't know, end of the year probably. Then the extent visualization that the users would like to see as a result of our analysis, more nice figures. just yes. And we also want to provide more figures, but it's all hard to code. And we code as fast as possible. But right now, we actually have a proposed project to Elixir to create this interactive visualization tool for plotting different results from RNA-seq, like heat maps, PCA plots, and so on. And the main part is the interactive, so then the user can actually play with it and see exactly what they want. And the last point, which was mentioned twice, was if we provide or will provide Oxford Nanopore or in general long read data analysis. And the truth is that I know it's big interest right now in the world, and I would be really interesting in have more experience in long read analysis, but so far there were not actually much of a demand here. I Philip had some experiments to data, but there were no actual big project that would demand us to work with long reads. So hopefully, if there is demand, we definitely gonna, I guess, be up to the challenge. And there's the acknowledgement. We are unfortunately still not part of any infrastructure in Czech Republic, but we're going to be from 23. But right now, it's still just a normal acknowledgement. That's it. Thank you. And if you have any questions.
Yeah, thank you very much. So do we have some questions here?
Yes.
From your support to user, how would maybe it is seems that there is a lot of development. Do you have it? Is it 100% of development, or it's the projects are somehow standardized and the user can do it on their own once you have the pipeline built?
In theory, the users might do the computation of the standard things, do it themselves, but in practice, it's not a reality. From my experience, the biologists are really not going to write the command line, even if it's prepared, they're not going to do it. Actually, it touches on a subject which I maybe intentionally not mentioned in my presentation, that we had the Galaxy server of our instance of our Galaxy server running, which is still there, but we somehow discontinued the full support for it because it was not used.
What is Galaxy server?
Galaxy server is like a graphical interface for the pipeline. So you can have the pipeline, and then you just can select different algorithms graphically, like put them together, and then run it. But still, it was too much. Like, since we provide the service directly, they better ask us to do it, I would say.
Maybe would it be possible to edit that education part of your activities, that Galaxy server? Or
as I said, when we started this project, I'm not saying that we had a big hopes, but we invest time to actually run it, but we also invest quite a lot of time to educate the users to use it. And we had, I would say, 4 or 5 workshops for different research groups, even direct interactions during 2019 mainly. But then at the beginning 2020, still the throughput of the server was like 3 users a month or something, really, really low. So basically, except of several plant people, nobody were using it. So then we actually were not trying anymore. And because on the other hand, like the increase in people asking our service was still increasing, but the usage of the Galaxy server was decreasing. So we just focused more on our own pipelines and our own system to actually run it because it was demanded.
Well, I will just comment because some of the, it's not full analysis, but at least QC control, quality control, this kind of thing. And is when alignment is a standard part of this quality control, the alignment of the data is part of the primary analysis and quality control, this kind of thing. So it's provided to the users. And then it's the analysis, it's not so easy, I would say, and it's better to do it is at least supervised by experienced.
That's probably the main part why they don't work with. Primary analysis we do anyway, and secondary analysis is already not 100%, but a bit project dependent, and it's better if it's supervised by someone experienced. Yes.
Curious, so this educational part where you said you are like giving these courses now this autumn, right? You said this is like for what staff, or is it only for students? Or
These courses are as part of like Masaryk University courses. I have no idea if anybody can.
Okay, no, because you mentioned it, so I thought like you are somehow responsible for it because.
Yeah, I am, but the thing is that I have no idea how to, go to, or I think you can come. Just
it's standard for pre-grad and postgrad students. So
yes,
part of the setup for the students. And to be honest, I don't know how these classes are marketed to employees.
Yeah, well, the postdocs are.
Yeah, because,
I think you.
I don't know.
Yeah, if they are not marketed to you, you need to ask the teacher if you can attend. Usually,
you are not doing the presence. So
yeah, you do the outside.
I mean, okay, but I'm quite new here and I'm still not really getting used to all
Sometimes
the administrative issues, so it's not so transparent, like where you can register it or not. And, it's why I was just only thinking if you do this type of things, I guess you said you were doing workshops for Galaxy, right? And this didn't work out because, as you said, the primary analysis alignment. Well, if you anyway align, what's the point if I align myself? If I want to then do something a little bit more advanced, and anyway, I have to pop up at your room again. Yeah. Just in general, like for because you were pointing out this visualization thingies, like kind of repetitive course for R, ggplot, and statistical research.
Yes, that's one thing I'm really want to do each year, and right now there's this online course which needs to be actually being created by the end of the year, and from there I want to go soon. From next year, I hope I will at least once a year have this course for our usage for post-processing of RNA and DNA data, for post-processing NGS data, some statistics and visualization. Because I think, even though I might be naive, but I hope that at least some people who might be a bit experienced might get it and actually use it.
I mean, it's not so naive. I mean, the issue is like, as you said, biologists, when they use it for a publication, they use it once and they don't use it the next 3 years, and then when they get back to it, it's over. I mean, if you don't do it every day, recapitulating all the scripts and logic is quite a complex issue. But yeah, if you want to do it in the online course, it would go really great. It's just, well, I guess you described this as a Galaxy. Okay, that I understand the reason, but sometimes Galaxy is pretty straightforward. All the other things are pretty complicated. I mean, it's like, I don't know that people that do R programming, most of them are even capable to make a meta chain or something like that. To really make a good analysis. So it's kind of.
It's basically the complicated thing there. I'm sorry if I'm taking time for it, but the complicated thing there is to strike the balance between learning the basics and then just prescribe the formula how to, for example, create some specific plot. Because we can have the prescription of the formula how to create a specific plot, but then if you want to modify it, you're lost anyway. But to build all the skills from bottom to up is not possible anyway also. So it needs to strike a balance somewhere between and I'm still trying to from my experience, get into some nice shape this course, but I hope they're going to be the one soon.
I was just only thinking, if you have connections to Amble and to Vladimir and Co, just ask him whether, I mean, they have very advanced courses for idiots, right? I mean, like, you're like, unless you are a programming idiot at the start. And they have like different, like, how would I call it, clusters. So, you know, for the beginners, for the more advanced, and for the guys who really get into it. And you always have the possibility to get the material, download it, you know, you have all the cheat sheets and so on. They prepare it, it's there, it's never lost. So, you know, even if you took the course 3 years before and then you need it, you get into it very quickly. If you can, and something like that would be existing here. I think this would help a lot. And it would help you because it would relieve you from a lot of work.
I'll ask him when I'll be there.
Yeah.
Yeah, just ask him how they do it because it's really interesting how they have it set it up in the bioinformatics facility, especially there. It's kind of, you have always access to the material from the courses.
Yeah, I can agree. I was there.
It is
So
amazing, right?
yeah.
I mean, even though you have you forget the things, you still get back